Okay, so last time I went through the basically the catabolism, that is the breakdown of fatty acids. I talked about both saturated fatty acids and I talked about odd carbon fatty acids. I haven't talked about unsaturated fatty acids. I'm going to briefly talk about that. I find that when I talk about unsaturated fatty acids that people spend an inordinate amount of time worrying about it. Okay? Well, it's a little bit more complicated, so I understand the worry. But here's where I think it's important to see the big picture. And I'm going to tell you before I show you the big picture. And the big picture is that there are two additional enzymes that are involved in oxidizing unsaturated fatty acids. Just two. Okay? And they do interesting and important things. And I'm going to show you that. So here is what happens during oxidation of unsaturated fatty acids. Now, I don't even care that you be able to draw all the various structures that happen with these, because that's not the important thing. I do think you should know the names of the two enzymes, and I think you should know a little bit about what they do. Okay? Well, let me explain the problem to you first. The problem is that when we go through fatty acid oxidation, we've talked about oxidation of saturated fatty acids. That is, they had no double bonds in them whatsoever. But now we have to oxidize fatty acids that have double bonds, and you say, well, so what? We had intermediates in the process of oxidizing saturated fatty acids. We had those intermediates where we pulled the hydrogens off, and those were double bonds. Well, those double bonds were trans, and the double bonds that we find biologically are largely cis. Okay? So there's one issue that we have to deal with. Now, before I deal with that issue, I want to say a word about trans fats, because trans fats are relevant at this point, and they're not what you think they are. Okay? So trans fats arise not as a result of oxidation. This oxidation process that generates the trans intermediates is not, underline not, the source of trans fat. When people say trans fat, they're talking about fats that have unsaturated fatty acids in them, but that instead of having cis bonds, they have trans bonds. So the first question is, how do those trans bonds arise? The trans bonds in trans fat arise as a result of food processing. They arise as a result of action to partially hydrogenate unsaturated fats. Partially hydrogenate unsaturated fats. What does partial hydrogenation mean? I think I've talked about it here just a little bit before, but I'll just say it again. Partial hydrogenation means that if you have a fatty acid that is polyunsaturated, meaning it has more than one double bond in it, you're trying to add enough hydrogen to it to remove at least one double bond. Okay? Well, interestingly, what happens with biological fatty acids that have cis configurations and they have more than one double bond, what happens is when you add hydrogen to that, if you completely saturate it, that is, if you could add enough hydrogen, so not that you're going to partially hydrogenate it, but so that you're going to completely hydrogenate it, if you did that, you would make a saturated fat, because there wouldn't be any double bonds left. They're trying to make partially hydrogenated fat. They want to leave some of those double bonds back, and there are some reasons to do that. Okay? Remember, one of the things that double bonds does is affects melting point, and melting point does affect things like how stable a fat is and the melting temperature. Okay? So the food manufacturers will partially hydrogenate to keep some of those double bonds there. Well, what we've discovered over the years is that that partial hydrogenation that's done actually converts some of the cis bonds to trans bonds. That's what gives rise to trans fat. Trans fat is linked to some very nasty things. Coronary artery disease, atherosclerosis, increasing levels of LDLs. We'll talk about LDLs probably on Monday. Okay? But LDLs are what your doctor will call the bad cholesterol. So trans fats are associated with some very bad things. And so they become very much in the public eye in recent years. And as nutritionists, I'm sure you know about them and you've heard of them. I hope that you also know that just because a product says no trans fat, that doesn't mean no trans fat. Okay? No trans fat means less than 1 gram per serving. So we're going to have 0.9 grams of trans fat in that serving and still be able to call itself no trans fat. Okay? So that's important to understand. So when you look for, does this thing have trans fat or not, you don't look for the label that says no trans fat. You look on the back, and if you see the words partially hydrogenated blank, it's got trans fat. Okay? That's what you want to be thinking about. So if you see those words at all, that's an indicator to you that you're eating something that has trans fat in it. All right. So the trans fatty acids I'm talking about here have nothing to do with that. They are intermediates in an oxidation process. They do not make trans fat, period. Okay. Well, here we have cis. We've got cis bonds, and one of the things that we have to do in the process of oxidizing unsaturated fatty acids is we have to convert those cis bonds to trans bonds in the right place. That's the key. In the right place. Okay? If you recall last time when I went through the oxidation process, I showed you that all of the action occurred between carbons 2 and 3. Now I'm going to show you a simple situation to start where the double bond is not between carbons 2 and 3, and it's not a trans bond. All right? It actually occurs right here. So here's an oxidation that we started of oleic acid. Oleic acid has 1 double bond, and it's shown right there. This fatty acid can be oxidized by the regular beta oxidation process through 3 cycles. That is, we can knock off 3 acetyl-CoAs before we get to something that has to be converted. That conversion involves the first of those 2 enzymes that convert cis bonds into trans bonds of the appropriate nature. Okay? The first enzyme is called enoyl-CoA isomerase. What does it do? Well, it's very simple what it does. It takes a bond that is between carbons 3 and 4, 1, 2, 3, and 4. It takes a cis bond between carbons 3 and 4, and it converts it into a trans bond between carbons 2 and 3. Why does it do that? Well, this trans bond is a normal intermediate in oxidation of saturated fatty acids. So this guy can have water added to it, it can have the hydroxyl oxidized, and then beta oxidation can continue. So this one enzyme is taking something very simply that won't work and making it into a substrate that will work. It's very simple. This is the simpler of the 2 enzymes. Okay? Any questions on that? Yeah? So the trans is between C2 and 3. Okay? So all of the action in beta oxidation, and what this has made is an intermediate in beta oxidation. All of the action in beta oxidation is between carbons 2 and 3. Okay? All right. Well, that's the simpler of the 2 situations. There's one more complicated situation, and this is where students really start dwelling on it, and I don't think we want to dwell on it too much, but I'll just show you what's up. Here is linolenic acid. Linoleic acid, which has 2 double bonds. Linolenic has 3, but this has 2 double bonds. Okay? And we go through the regular oxidation process, and we get down to here, and what do we see? Well, we've got a cis bond between carbons 1, 2, 3, and 4, just like we had before. That one we can handle very easily because we've got enoyl-CoA isomerase, which converts a cis 3,4 into a trans 2,3. Very simple, just as we did before, and oxidation of that continues. All right? Then we get down here, and now we have a different scenario because what we have is a bond that's out here between carbons 1, 2, 3, 4, and 5. All right? This guy can start the oxidation process, but it can't finish it. Well, what do I mean to start the oxidation process? And starting the oxidation process means I make a trans double bond between carbons 2 and 3, just like I normally would in beta oxidation. But now I've got a double bond here, and I've got a double bond here. And chemically, the enzymes will not handle that. So we need a different enzyme to convert this guy into a substrate that's useful. So this guy has two double bonds. They're between carbons 2 and 3 and between carbons 4 and 5. The next enzyme, which is the other enzyme that's involved in oxidation of unsaturated fatty acids, is known as 2,4-dienoyl-CoA reductase. Mouthful of a name. Would you like to name it? By the way, the TAs on the last exam, I didn't tell them about Bob. So they figured it out. They came to me and said, it took me the longest time to figure out what the hell everybody was talking about with Bob. And they had never had me as an instructor before, so they didn't know I did this. But they figured it out, so I was kind of cool. I was kind of pleased with that. That was kind of cool. Anyway, so I think we should name this enzyme. What should we call the enzyme? Stacy? I have a vote for Stacy. Is it Stacy? Stacy? Do you have a name?
No, what is the name again?
Well, the actual name is 2,4-dienoyl-CoA reductase.
Dina.
What's that?
Dina.
Dina is even simpler, okay? Dina and Stacy. Female names are dominating this one. Huh? Brian? we may get in some trouble with that one, I think. So we have Dina, we have Stacy, and we have Brian. Dina? Stacy? Brian? Okay, I think it's Dina. All right, so we should call this enzyme Dina, which is not bad because dienoyl sounds kind of like Dina anyway, right? All right, so what does this enzyme do? Well, it takes those two double bonds, it takes some electrons from NADPH, and then it magically makes them into the substrate that is almost needed, okay? And in fact, this should actually be a cis bond, not a trans bond, plastic. This should be a cis, all right. So it makes it into a 3,4 cis. What happens to a 3,4 cis? A 3,4 cis is converted into a 2,3 trans by our same enzyme as before. So these two enzymes working together can oxidize any unsaturated fatty acid. What's the big picture? That's the big picture. Those two enzymes can do that. All right? So as I said, I'm not interested in having you draw those structures because I don't think drawing those structures tells me anything about you understand what's going on. I think you should know that the enoyl-CoA isomerase is the simpler of the two. And I think you should know this guy here is the more complicated of the two. But the sum of these two is that they convert the substrate into something that can be oxidized in beta oxidation. With these two enzymes, the cell is set to go. Questions about that? Yes? The two bonds? Okay, so the bonds are, there's a trans and a cis. The trans is between 2 and 3. The cis is between 4 and 5. But again, you're not going to draw that, so I'm not asking you to draw that. Okay. Let's move on and talk about a topic that's increasingly in the news and one that is interesting, although it's a sort of a sidelight, I'll be honest with you, with respect to most metabolism. All right? There's a lot of talk about ketogenic diets and things like that. So I want to say something about what those are and what that means. So ketogenic diets are those which give rise to production of ketone bodies. And ketone bodies are something I'll talk about here. They're actually shown down below. All right? This being one of the primary ones right here, beta-hydroxybutyrate, which isn't even a ketone. Okay? But these diets give rise to ketone bodies because they are glucose limiting. They are glucose limiting. Now, there's some interesting things that people are finding with ketone diets. Okay? They don't affect most people, to be honest with you. Okay? But they do affect, interestingly, kids with epilepsy. Okay? So it has been found in recent years that ketone diets for kids with epilepsy can have an enormous effect on reducing the likelihood of epileptic seizures, at least for some types of epilepsy. And kids who have these epileptic seizures and suffer from this get put onto diets that have essentially as little carbohydrate as they can possibly have. They're very rich in fat. There are things that we would think of are really diets that are not very good for you. For a child, that's less of an issue. All right? But they're very rich in fat because that's the primary source of energy, and they're very, very limiting in carbohydrates that are there. All right? And they're called ketogenic because when you run out of glucose and you have little in the way of glucose stores, your body starts making ketone bodies as a way to keep the brain and your eyes functioning. That's the primary things that these guys do. All right? Well, what happens? Why do we have them and so forth? Glucose, as you have seen, is a source of immediate energy. It's also a very important energy source for the brain and for your eyes. Okay? People who go on starvation diets, you hear about people who go on a fast, to protest something, that may go and stay on that diet until literally they die. What happens is before they die is they go blind, okay? Because their eyes are needing glucose. And when you're on those kinds of diets, your body runs out of resources to make glucose. So in an effort to keep the brain and the eyes alive, the body uses alternative energy sources for the brain and for the eyes that don't involve glucose. So normally we're circulating glucose in our bloodstream for that purpose, because our brain can't make glucose. It's dependent upon what our liver does. And one of the reasons, even though glucose is a poison, one of the reasons our body is making that glucose at a constant level in our bloodstream is to keep the brain going. Very, very important. Brain's an important consumer of glucose. Well, when glucose isn't available, all right, and you know that you're an animal, and I told you before that animals can't make glucose from fat or fatty acids. Okay? We can't do that. Plants and bacteria can do that. Okay? It's nice to be able to use fats for energy to keep the brain going. Well, how, if you don't make glucose, do they do that? Well, they make ketone bodies. You've seen that oxidation of fatty acids gives acetyl-CoA. So when you're on one of these diets where you have very limiting amounts of glucose available, all right, what you are producing is a lot of acetyl-CoA. The reaction I'm going to show you here is how we make ketone bodies, and we will see that it's also involved in making cholesterol. At least part of it is. Okay? Well, how does it work? We start with two acetyl-CoAs. We put them together and we make this 4-carbon molecule. I don't care if you know the name or not. All right? 2 carbons plus 2 carbons gives 4 carbons. And if you notice the name of the enzyme up there, you'll recognize that that was the last enzyme of fatty acid oxidation. That's an important enzyme name. A very important enzyme name. Thiolase catalyzes the breakdown, if we go upwards, the breakdown of this, splitting it in half to make 2 acetyl-CoAs. Or if we have an excess of acetyl-CoA, the same enzyme catalyzes the joining and making of this acetoacetate-CoA. So abundance determines the direction that thiolase catalyzes this reaction. When we have plenty of acetyl-CoA, it's going to move downwards. When we're burning up our acetyl-CoA, it's going to move upwards. Okay. The 4-carbon molecule gains another 2-carbon molecule to make a 6-carbon molecule, which has a mouthful of a name, beta-hydroxy-beta-methyl-glutaril-CoA, and we would give it a name except for the fact that that's already been done. It's called HMG-CoA. Okay? HMG-CoA turns out to be an intermediate both in the synthesis of ketone bodies and in the synthesis of cholesterol. So this guy can go to the right to another pathway to make cholesterol. We'll talk about that later. In this pathway, HMG-CoA loses a 2-carbon piece and becomes a different 4-carbon piece. This was acetoacetyl-CoA. This is the same thing without the CoA. It's called acetoacetate. This is a ketone body. Acetoacetate is a ketone body. Well, it's not a real good ketone body. And the reason it's not a good ketone body is that it's somewhat chemically unstable. All right? It will spontaneously give off carbon dioxide at a low frequency. And when it does, it produces this guy over here, acetone, which is totally not useful for the body. Your body can't metabolize acetone. All right? So to keep that from happening, the body will often reduce acetoacetate and make beta-hydroxybutyrate. This is much more stable, and it travels in the bloodstream more readily. We call it a ketone body, but it's not really a ketone body because there's no ketone in it. Okay? And again, that should be an O up there, but that's missing an O. All right. Now, when this guy travels through the bloodstream and gets to the brain, this process reverses. This guy goes to this guy, goes to this guy, goes to this guy, and finally goes to acetyl-CoA. And the brain can use that acetyl-CoA in the citric acid cycle and make energy. So what the body has done is it's taken something from fatty acid oxidation and used it in a delivery mechanism to get it to the brain so the brain can have energy that doesn't come from glucose. Okay? Does that make sense? Yes, sir. So the brain, when this gets to the brain, this whole process goes backwards and makes acetyl-CoA. That's correct. So in the liver, it's going to go this way, dumps in the bloodstream, and then in the brain, it's going to go back up this way. Okay? Now, a couple things here. One thing here that's important is this is a dead end. Your body can't metabolize acetone. So what happens is it gets to your lungs and you exhale it. Acetone is fairly volatile. It's an organic solvent. You use it for fingernail polish remover. That's what it smells like. Okay? And this is an important health consideration because people who are diabetic will frequently get very hypoglycemic, meaning they have very low blood glucose levels. What happens when you have very low blood glucose levels? This process. And as this process goes, one of the byproducts is acetone. All right? If you ever smell acetone on somebody's breath, you want to tell them to get to the doctor because they may very well have diabetes and not know it. I usually have about 1 per year where somebody discovers this. Right? If you have a friend who you smell acetone on their breath, there's a good chance that they have diabetes. Okay? Acetone smells like fingernail polish remover. Okay? And it's happening because, again, the blood glucose level has gotten so low, the body says, I better be dumping something out here to keep the brain going, or we're going to have trouble. And that's what's happening with that. Okay. That's the last, I will say, of fatty acid metabolism there. Yes? So when it's in the brain, it moves up. And when it's being made in the liver, it's going down. That's correct. Okay? Yes? Where? Any cells that need it. Yes, it will. Yep. Okay? Okay, well, why don't we sing a song to celebrate the end of fatty acid oxidation? And it's to the tune of the song When Johnny Comes Marching Home. Okay? The fatty acids carried by CoA are oxidized inside the mitochondriae. They get to there, as you have seen, by hitching rides on carnitine. Then it goes away when acids get oxidized. Electrons move through membranes. Yes, it's true. It's true. They jump from complex 1 onto CoQ. CoQ. The action can be quite intense when building proton gradients, and it's good for you when acids get oxidized. The protons pass through complex 5, you see. They do this to make lots of ATP. The mechanism you should know goes through the stages LTO, so there's energy when acids get oxidized. Okay, let's see. I'll use that in a minute for something else. Okay, so let's now turn our attention to fatty acid synthesis. I told you we're going to have a lot of metabolic pathways, and we do. Now, the very good thing about fatty acid biosynthesis is that the reactions themselves are very much like the reversal of fatty acid oxidation. There are some important differences, but they're very, very similar to fatty acid oxidation. All right? Well, first of all, fatty acid biosynthesis occurs not in the mitochondrion. Remember, oxidation and all this is going on inside the mitochondrion. Fatty acid biosynthesis is going on inside of the cytoplasm. Going on inside of the cytoplasm. All right, now what does that mean? It means that if we are, let's say we're eating a lot of glucose, and we are making all kinds of acetyl-CoA because we're getting all that pyruvate, we're getting this pyruvate in here, and we're getting this acetyl-CoA being produced, but we're not exercising. What happens when we don't exercise? Well, we went through that whole process of respiration and respiratory control the other day. The bottom line was if we're eating more than we're exercising, what's happening is we're going to stop the citric acid cycle because we're going to have too much NADH and not enough NAD. The citric acid cycle is going to stop. Well, if we keep eating and we've got more sugar than we should be eating and we're getting all these carbs in here, our acetyl-CoA concentrations start increasing. Our acetyl-CoA concentrations start increasing. Now, I'd like you to think for a moment to that citric acid cycle. You may or may not remember this, but I told you that that reaction between oxaloacetate and acetyl-CoA was energetically very favorable. Very, very favorable. I said it was favorable enough it actually pulled the preceding reaction. Well, this being very favorable is important because as acetyl-CoA concentration goes up and we have oxaloacetate present to deal with that, we can convert that into citrate very easily. All right? If we convert citrate to isocitrate, we can do that, but we don't really do that very much because isocitrate accumulates because it can't be oxidized in the next step. So the bottom line is we can make a lot of citrate, but we can't do anything with it. We can't do very much with it because the following reactions after that are requiring NAD and they get blocked. All right? So citrate starts to accumulate. Well, that turns out to be the #1 signal to make fat. There's two reasons why. First of all, citrate can be transported across the mitochondrial inner membrane. It can be transported across the mitochondrial inner membrane, and when it gets out into the cytoplasm, an enzyme will cleave off oxaloacetate and acetyl-CoA. We have just shuttled acetyl-CoA out into the cytoplasm. Citrate is a shuttle to carry acetyl-CoA out into the cytoplasm. Now, I said citrate affects this in two ways. One is it's carrying acetyl-CoA out into the cytoplasm. The second way, we will see in just a little bit, but it's going to stimulate the first enzyme in the synthesis of fatty acids. It stimulates the first enzyme in the synthesis of fatty acids. So citrate's going to nail you in two ways, if nailing you means that you're making too many fatty acids. All right. So we've now gotten acetyl-CoA out into the cytoplasm. All right. We're ready to start making a fatty acid. When are we going to make fatty acids? We're going to make fatty acids, first of all, when we have a lot of acetyl-CoA in the cytoplasm. We're going to make fatty acids when we have a lot of ATP energy. And we're going to make fatty acids when we have a lot of NADPH or NADH. All of those are going to be conditions that are going to give rise to the synthesis of fatty acids, and all of those are going to happen when we're eating more than we are exercising. All right. Well, the first step in the process is this reaction right here. It's an odd reaction, and it's a very interesting one. The enzyme that catalyzes this has what sounds like a mouthful of name, but it's actually quite easy. The name of the enzyme is acetyl-CoA carboxylase. Acetyl-CoA carboxylase. As I've told you before, when you hear the term carboxylase in an enzyme name, it means biotin is a coenzyme, and there's biotin, because biotin is very useful for putting carboxyl groups onto things. And that's what this enzyme is doing. We're starting with a 2-carbon molecule. We're using ATP energy, and we are making a 3-carbon molecule as a result of putting on a carbon dioxide, or bicarbonate, same thing. Okay? This guy right here, malonyl-CoA, will be slightly altered in the next step, and then we're ready to start making fatty acids. Okay? All right. Now, this enzyme acetyl-CoA carboxylase is regulated in 2 ways. I've just told you one. Actually, 3 ways if you want to count them. One is that it's stimulated by citrate. This enzyme, as citrate concentrations are high, this enzyme gets activated. Okay? This enzyme is inhibited allosterically by the end product of fatty acid biosynthesis. And as we will see, that end product is palmitic acid. So it's activated by citrate, it's inactivated by its end product, palmitic acid. And you might say, hey, that's great, it stops me from making too much fat. It doesn't, because palmitic acid gets put into fat, and fat doesn't inhibit this enzyme. So only when palmitic acid is accumulating does this enzyme get inhibited. Okay. Now, where am I at here? That's the mechanism of the reaction. You don't need to know the mechanism. We won't go through that. The synthesis of palmitic acid is surprisingly simple. Now, first of all, let's talk about palmitic acid. It is a 16-carbon fatty acid. I just described it as the end product. You say, well, we have longer fatty acids. It's the end product of this cycle I'm showing you here. And this cycle is one of the ends of fatty acid biosynthesis. If we go beyond 16, if we want to make a fatty acid that has 18 or 20 or 22 or 24, that's a different process, okay? And it's located in different places, okay? So for this process, which is occurring in the cytoplasm, palmitic acid is an endpoint. Well, what happens here? Well, here's an acetyl-CoA, okay? Here is a malonyl-CoA. To start the process, I have to have one of each. An acetyl-CoA, I have to have a 2-carbon piece, and I have to have a 3-carbon piece. The first thing that happens to both of them is that they both have their CoA swapped out for something called acyl carrier protein, or ACP. So instead of having acetyl-CoA, I end up with acetyl-ACP. And instead of having malonyl-CoA, I have malonyl-ACP. Okay? It's just a different thing holding on to the individual group. This, one way we can tell that synthesis is going on is the fact that ACP will be attached to it. Okay. Once we've gotten these guys together, once we've gotten these guys made, that is the acetyl-ACP and the malonyl-ACP, we join them. And when we join them, this carbon dioxide that we put on pops off. So in essence, we're taking a piece that has 2 carbons, a piece that has 3 carbons, and we're making a piece that has 4 carbons, and we're losing a carbon dioxide. So 2 carbons plus 3 carbons gives us 4 carbons plus a carbon dioxide. The carbon dioxide goes away. We don't care about it. There's a 4-carbon piece. Okay? Now, this looks a little bit like ketone body synthesis, except for we used a 3-carbon intermediate instead of a 2-carbon intermediate. All right? The next step in the process are just going to be the reversal of the reactions we saw for fatty acid oxidation. The first thing that happens is, notice that we have a ketone on carbon number 3. So the first thing we do is we reverse that ketone backwards into a hydroxyl group. No, you don't need to know the name of the enzyme. The second thing that happens is we remove water across here to make a double bond, and we make a trans double bond just as we did before. The next thing we do is we add hydrogens and electrons across there to reduce that completely, and now we've got a 4-carbon piece. Okay? Then this guy comes up here, and instead of having a 2-carbon piece, I have a 4-carbon piece that now gains 2 more from another malonyl-CoA. So this process cycles over and over and over until we end up with 16 carbons. Now, it turns out that this process occurs not with individual enzymes as you see labeled here, but instead all of these enzymes are together in one complex. And this is really simple. Okay? The one complex has a simple name. It's called fatty acid synthase. It doesn't get any better than that. The complex is found in organisms all the way from bacteria to human beings. And it's known to also be one of these rotating enzymes. It takes and it grabs the things and puts them over in one place where the first reaction occurs. It moves it down to 4 o'clock where the next reaction occurs, moves down to where the next reaction occurs, and goes all the way around. When it's gotten all the way back up around, instead of having that 2-carbon piece, it's now got a 4-carbon piece. The next time around, it's going to have a 6-carbon piece. The next time around, it's going to have an 8-carbon piece, etc. And by the time it gets finally to a 16-carbon piece, it's done. When it gets to the 16-carbon piece, palmitic acid, it lets go of it, and the process is completed. Okay? Yes? Will it ever start from a 4 or 6-carbon piece? That's a good question. I don't know that it will. I think it will always start with an acetyl-CoA, but I don't know for sure if that's the case. I just don't know. I would suspect it would always start with a 2 because I think getting a 4-carbon piece in there, it probably wouldn't load properly, and a 2-carbon piece would, but I can't tell you that definitively. I don't know. All right. We have just made this guy. All right. What do I want to say? Palmitic acid, coenzyme A. Oh, yeah, okay. This is kind of cool. All right. You might say, well, why do they have ACP? And I'm not even sure why cells use ACP. ACP is a protein. It's called acyl carrier protein. But the thing that's sticking off of the protein is almost identical. In fact, it is identical to the thing that's sticking off of coenzyme A. So this handle is very, very similar. Here we have a protein. Here we have basically AMP that's linked to that. Okay? That's what that is. All right. You guys are tired. I'm tired. I would propose we sing one more song, and then we call it a week. You like that? All right. So the other song is called When Acids Are Synthesized. And it has exactly the same tune. And we don't sing it backwards, okay? So, the 16-carbon fatty acid palmitate gets all the carbons that it needs from acetate, which citric acid helps release from mitochondria matrices. Oh, a shuttle's great when acids are synthesized. Carboxylase takes substrate and it puts within. Dioxycarbon carried on a biotin. CoAs all gain a quick release replaced by larger ACPs, and it all begins when acids are synthesized. A malonate contributes to the growing chain. 2 carbon 7 times around again. For saturated acylates, there's lots of NADPH that you must obtain when acids are synthesized. Palmitic acid made this way all gets released. Desaturases act to make omega-3s. The finished products, big and small, form esters with a glycerol, so you get obese when acids are synthesized. See you guys on Monday.